Good morning, it's November 26th, and this is your daily brief in science. Here's everything you need to know. The Philippines is placing radiation technology at the center of its strategy to cut plastic pollution, with the IAEA's New Tech Plastics Forum showcasing how radiation processing can upgrade low-value plastic waste into durable materials. A highlight is the Post-Radiation Reactive Extrusion Project, or PRX, which aims to turn waste plastics into usable products, and includes a PREX prototype house in Quezon City to demonstrate applications for community housing. Since its 2021 launch, IAEA New Tech Plastics has supported more than 100 member states in tracking microplastics, identifying plastic types, and applying harmonized lab methods while promoting radiation technologies for practical materials. President Marcos has urged that technological growth align with human well-being, ethical standards, environmental integrity, and sustainable development within a global tech shift. The IAEA, established in 1957, works to promote peaceful uses of nuclear energy, and the forum runs through November 26, 2025, with technical sessions, exhibitions, and discussions to foster international collaboration in environmental protection. Grossi emphasized awareness of new nuclear technologies and small modular reactors, noting Marcos's strong knowledge on the topic and directing technical advisors to inform decision-makers. The forum agenda centers on a circular economy through radiation technologies, enhanced microplastics monitoring for coastal policy, and partnerships to drive sustainable development. The IAEA aims to help the Philippines identify suitable nuclear energy systems beyond traditional power plants, including SMRs, and a new agreement with the Asian Development Bank strengthens regional financing and technical support for nuclear power projects. The Philippines has rejoined the IAEA Board of Governors for 2025 to 2027, reinforcing its role in shaping global nuclear policies and expanding technical support in nuclear medicine, agriculture, food security, and energy, as the two-day event highlights cross-sector collaboration and leadership in applying nuclear and radiation technologies for practical solutions. A UC Davis-led study finds TIGIT behaves differently in rhesus macaques than in humans, a result that could change how we evaluate TIGIT-targeted cancer therapies. The work shows plasmin exposure causes macaque TIGIT to shed from immune cells, creating a soluble form that can still bind anti-TIGIT antibodies like tiragolumab, potentially diluting the drug's effect. Researchers engineered both human and macaque versions of TIGIT and found the macaque protein is shed by plasmin, while the human version is not. This species difference hinges on a single amino acid change at position 119. In humans, TIGIT remains on the cell surface under the same conditions, but in macaques it becomes a soluble, bindable target. The finding raises questions about the translational relevance of macaque data for human trials in TIGIT-targeted therapies. Anti-TIGIT antibodies have had limited success in Phase 3 solid tumor trials, prompting a closer look at underlying biology and model systems. The study notes that the plasmin-mediated release of soluble TIGIT could soak up therapeutic antibodies, altering apparent efficacy and safety profiles in preclinical testing. Context from the research community emphasizes that improving human-relevant models is essential to accurately predict human outcomes. The team, led by Joginder Tushar Singh at UC Davis with collaborators including Eric Perillo, received NIH/NCI support and backing from the Ovarian Cancer Alliance of Greater Cincinnati. Overall, the work suggests that macaque models may misrepresent human safety and dosing for TIGIT therapies, underscoring the need for caution in translating preclinical findings to humans. Urban habitats in the Keihanshin region are shaping a plant's evolution, with traits like height and flowering period diverging across farmland, parks, and roadside environments. The study notes an 8°C rise in maximum ground temperature in cities, along with artificial shading and soils that are less acidic, factors linked to adaptive trait diversification. Researchers conducted field comparisons in Osaka, Kyoto, and Kobe to understand how urban habitat diversity drives evolution in this species. The work, published in the Journal of Ecology in 2025, was funded by JST and JSPS with collaboration from the University of Hyogo and summarized by EuroCalert. Prominent researchers cited include Kobe University ecologist Ushimaru Atsushi and former graduate student Nakata Taichi, now at Kyushu University. Future work will use cultivation experiments that simulate field conditions to test trait benefits and to determine if adaptations are embedded in the plant's DNA. Follow-up studies will employ controlled yet field-like conditions to assess fitness advantages of different traits and to investigate genetic encoding of these adaptations. In summary, the researchers aim to show how cultivation under realistic conditions reveals divergent trait benefits and possible genomic engraving. This study is described as a starting point for broader research into how urban habitat diversity in megacities shapes plant evolution. The paper is titled Adaptive Trait Divergence of Annual Plants in Response to Urban Habitat Diversity in a Megacity and appears in the Journal of Ecology, 2025. The research presents evidence of rapid adaptive evolution occurring in roughly 6 decades due to urbanization and argues that founder effects alone do not explain the divergence observed. A tiny protein with a big role could reshape how we think about fat storage. A microprotein named adipogenin, comprised of 80 amino acids, promotes lipid droplet formation and healthy lipid storage in fat cells. UT Southwestern and Helsinki researchers used advanced imaging to show adipogenin binds mainly to the sapin complex, reinforcing its structure to stabilize the fat storage machinery and drive lipid droplet growth. Cryo-electron microscopy indicates this interaction makes the sapin complex more rigid and stable, enabling the formation of larger lipid droplets and greater fat storage in adipocytes. The study used mice and high-resolution imaging to map adipogenin's role in lipid metabolism, with Chao Li and Shunan Sun as co-first authors and Elina Ikonen as co-senior author. NIH funding and foundations supported the work, which was published in Science and utilized UT Southwestern's cryo-electron microscopy facility. The findings point toward clinical applications by identifying a novel target to modulate fat storage and improve metabolic health. In practical terms, adipogenin could be targeted to dampen harmful fat buildup or enhance healthy adipose storage, with broad implications for obesity, diabetes, lipodystrophy, and fatty liver disease. Overproduction of adipogenin in mouse models led to larger lipid droplets and increased fat storage, while deficiency produced smaller droplets and less fat. The research underscores the broader clinical relevance of controlled lipid storage to metabolic health and disease risk, marking adipogenin as a potential druggable lever for modulating fat storage. A new study highlights how MAPK-driven epithelial plasticity fuels regenerative stem-like states in colorectal cancer cells and links this to therapy resistance. In vivo, oncogenic MAPK signaling from KRAS mutations, roughly 40 to 50%, and BRAF mutations, about 10%, triggers epithelial state changes, while MAPK inhibition rapidly reprograms transcription to push tumors toward a canonical WNT-associated stem phenotype. The findings point to a potential shift in therapy design, suggesting treatments that manage epithelial plasticity alongside MAPK blockade for more durable colorectal cancer responses. Understanding when resistance and plasticity arise helps identify optimal treatment windows, especially in tumor stages with lower intratumoral heterogeneity. Clinically, exceptional responses have been observed with combined BRAF and EGFR therapies in BRAF/RNF43 co-mutant tumors, underscoring plasticity's role in outcomes. Detailed observations of these exceptional responses can be explained by restricted plasticity, supporting integrated genetic and phenotypic profiling. Epithelial plasticity sits at the center of resistance. Restricting plasticity or targeting WNT pathway components, including RNF43-related ligands, can enhance MAPK inhibitor efficacy. Restraining plasticity early in metastasis or targeting ligand-dependent WNT mutations yields strong therapeutic responses. KRAS mutant tumors show acute resistance to MAPK inhibitors due to rapid plasticity and WNT program activation, while BRAF mutant tumors experience delayed resistance. The paths to resistance differ by genetic context, with KRAS-driven tumors reacting quickly and BRAF-driven tumors developing resistance more slowly, shaping how therapies are combined. Therapeutic implications include combining MAPK inhibitors with WNT modulators or stem cell regulatory axis agents to curb phenotypic switching for durable responses, and the study urges targeting cancer cell plasticity as part of CRC resistance management. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.